Welcome to this YouTube channel, and hope your subscription.
Nucleic acids. Nucleic acid is the most important molecule out of the main biomolecules in living matter in genetical aspect. It is a linear polymer made up of large number of nucleotides. It contains carbon, hydrogen, oxygen, nitrogen and phosphorus. Each nucleotide is made up of 3 components. They are nitrogenous base, pentose sugar group, phosphate group. There are 2 types of nucleic acids: DNA, deoxyribonucleic acid; RNA, ribonucleic acid. DNA. The structural unit of DNA is deoxyribonucleotide. DNA transfers genetic characteristics from generation to generation. RNA. Except DNA, the other nucleic acid present in organisms is RNA. The structural unit of RNA is ribonucleotide. Protein synthesis is the function of RNA. Significance of nucleic acids. Important in storage of genetic information of organisms. Important in transferring genetic information from generation to generation. Important in protein synthesis process. Important in controlling all cellular activities in a cell. The information to control cellular activities is present in DNA. RNA is important in storing genetic information of some viruses. The variations occur in DNA due to mutations are important in evolution. The above biomolecules contain mainly carbon, hydrogen, oxygen and nitrogen. We will conduct below mentioned activities to confirm the presence of those elements. Activity 1. Identification of water as a constituent in food. Materials required: meat, egg shell, plant leaves. Crucibles. Method: grind, crush meat, egg shell, and leaves in their dried form separately. Put them separately into the crucibles and heat them. During heating, hold a glass sheet above the crucible. Use anhydrous cobalt chloride, copper sulfate to identify whether the liquid drops on the glass is water. Blue-colored anhydrous cobalt chloride turn to pink and white-colored anhydrous copper sulfate turn to blue. Then it is confirmed that water is formed on the glass sheet. Therefore, the food that is used for the experiment contains water as a constituent. Activity 2. Identification of presence of carbon in biomolecules. Materials required: several crucibles. Spinach stems, piece of fish, chickpea. Method: make pulps by crushing all above materials separately. Put them separately into crucibles and heat well. The final residue obtained should be rubbed against a white paper. Lines drawn due to coal as observed can confirm the food that is used for the experiment contains carbon. Activity 3. Identification of presence of nitrogen in biomolecules. Materials required: 2 test tubes, solution of sodium hydroxide, solution of copper sulfate, egg white, piece of fish. Method: crush fish thoroughly, add water and mix well. Filter the solution. Put 2 milliliters of the fish extraction and egg white into separate test tubes. Add equal volume of sodium hydroxide. Add few drops of copper sulfate to it. Purple color appears in the solution and this confirms the presence of protein in food, as nitrogen is a constituent of proteins. It is confirmed that the above tissues contain nitrogen.
Hope your subscription. Thank you.